Hello student, welcome to Navshala. We provide quality education free of cost. No paid batches, no membership. Only free educations to everyone. Children, in last lecture we started the chapter Heredity and Evolution from class 10. In that lecture we discussed about heredity and central dogma. In heredity there is a transfer of biological character from one generation to another generation with the help of gene. And the second concept we discussed about central dogma, where we understood that all information about protein synthesis is present in DNA. So protein is synthesized by DNA through RNA is called as central dogma. So in last lecture we understood that if you want to make any protein, DNA is very important there. So in today's lecture we are discussing about what is structure of DNA, RNA and how it helps in protein synthesis. Let's start. Now in previous lecture we completed that DNA is a double stranded structure there. So DNA is made up of two strands. It is called double helix model given by Watson and Crick in 1953. So this DNA model consists two strands. Suppose this is the first strand of DNA and this is second strand of DNA. If you observe carefully here that each strand consists of repeating units. If you see here, this is one unit there. Somewhat same unit you will find below that. So this is the first unit there, this is second, third, fourth. Like that many small repeating units makes one strand of DNA. So what is that repeating units? These repeating units we called as a nucleotides. So this is your first nucleotide there, this is second nucleotide, third nucleotide. Children, how it is? If you join many small flowers, then one big mala will form. Somewhat same concept there here. Your flower, who is there? Flower is nucleotide there and the whole mala is your DNA there. So how mala is made up of small flowers, like that each DNA strand is made up of small units called as nucleotide. Are you getting? In mala small flowers are there, in DNA strand nucleotides are there. So if you remember, in flowers also some parts are there, no? Calyx, corolla, androecium, gynoecium, like that. So which nucleotide we are talking? No, in this nucleotide also some components are there, simple there. So each nucleotide is made up of three components. One is phosphate group, second is sugar and third one is nitrogen base. Each nucleotide is made up of how many components? Three components, which are that first phosphate group, second sugar and third is nitrogen base. Clear? Now here important thing you have to remember that the sugar which is present in DNA, what's its name? So the sugar present in DNA is called as deoxyribose sugar. So in papers such type of question they can ask which type of sugar is present in DNA, like that. Okay? So what will remember that DNA is a chain of nucleotide. Each nucleotide consists how many components? Three components. One, sugar, phosphate group, nitrogen base. Which sugar present in DNA? Deoxyribose sugar. Now these nitrogen bases which are present in DNA are of four types there. If you remember, in 9th standard also we discussed this: adenine, guanine, cytosine and thymine. So above this, starting three which are there, adenine, guanine, cytosine, it is present in DNA as well as in RNA. So you have to remember the fourth one. What is that? Thymine. Why, sir? Because thymine is a nitrogen base which is present only in DNA, absent in RNA. So which is special? We have to remember that. Clear? So here two questions related to objective. One, which is the sugar present in DNA? Deoxyribose. And which is a nitrogen base present only in DNA? That is your thymine. Is it clear? So what to remember about the DNA structure? Simple points are there. DNA double stranded there. It is made up of two strands. Each strand of DNA is made up of chain of nucleotides. Many nucleotides will join to make one DNA. Sir, this each nucleotide consists how many components? Three components. Phosphate group, sugar, nitrogen base. Which sugar present, sir? Sugar present in DNA is deoxyribose. Which four types of nitrogen bases are there? Adenine, guanine, cytosine and thymine. Starting three are present in both. Thymine is present only in DNA. Till here, any problem? Is it clear to everyone? Now let's understand the structure of RNA. RNA structure is also somewhat similar to DNA, only 1-2 points are different than this. Please understand the structure of RNA related to protein synthesis. Now children, important here, first thing which you have to remember that just now we discussed DNA double stranded there. So RNA is single stranded there. So first thing, RNA is not double stranded, it is a single stranded there. Second, if you compare DNA RNA by chain of nucleotide, here also present. It is also made up of repeating units. This is your first repeating unit there. Then this is the second one there. So 1, 2, 3, 4, somewhat same structure of DNA RNA there in terms of nucleotide. So DNA strand is also chain of nucleotide. RNA strand is also chain of nucleotide. Then the difference, what the difference in two cases? One, the sugar which is present in RNA is ribose sugar, where it is deoxyribose sugar in case of DNA. So here the sugar present is ribose sugar, remember this. Rest phosphate group here also there, nitrogen base here also there. So each nucleotide is made up of 3 components, phosphate, sugar, nitrogen base. Sugar present in DNA is deoxyribose and sugar present in RNA is ribose. Now let's see about the nitrogen bases. Just now we discussed that starting 3 are there, it is same for both DNA, RNA, 4th one is changing. In DNA there is a thymine, where in RNA it is uracil. Is it clear? So the uracil is present only in RNA, absent in DNA. Clear? Now let's compare both. What is DNA? DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Deoxyribonucleic acid, ribonucleic acid. First. Second, DNA double stranded there, RNA single stranded there. Third, which sugar present in DNA? Deoxyribose sugar is present in DNA, where RNA consists ribose sugar. Which is a nitrogen base which is present only in DNA? Thymine. So thymine is present only in DNA, while uracil is present only in RNA. Is it clear? Once again, DNA, deoxyribonucleic acid, ribonucleic acid, double stranded, single stranded. It consists deoxyribose sugar, it consists ribose sugar. Here thymine is present, here uracil is present. I think DNA, RNA structure and difference you understood. Now let's check how much your understanding happened about DNA and RNA. If I ask you, first question, that DNA contain which sugar? Whether it is ribose sugar present, deoxyribose, glucose or fructose. Pause the video and answer me in comment box. What is the sugar which is present in DNA? Good. Many students, 100% I am knowing, you will give the answer. Deoxyribose sugar is present in DNA. Second, if I ask you which is the nitrogen base, which nitrogen base is present only in DNA? We are having 5 total: adenine, guanine, cytosine, thymine and uracil. Which one is only present in DNA? Fast you have to comment. Correct. So the which nitrogen base present only in DNA? So your answer should be thymine, because thymine is present only in DNA. Now this question I want answer from everyone in comment box, that which nitrogen base is present only in RNA. This answer I will not tell. You will tell in comment box that in RNA which nitrogen base present. Only in RNA, which is absent in DNA. Is it clear? So up till now we discussed about in central dogma which DNA RNA helps, what is their structure and what are their differences. Now after understanding the structure of DNA and RNA, now we can move to the main process, that is protein synthesis. Children, in protein synthesis, the first step which we want to discuss, it is related to transcription. In transcription, what we have to understand that in central dogma we are telling whatever information is present about the protein synthesis, it is present in DNA. So DNA, what doing? DNA will pass its information to the second structure, that is RNA. So DNA's information, when it will go to RNA, RNA will take this information to one important unit called as a ribosome. Where this ribosome present? It is present in cytoplasm, and this ribosome finally will make our main important product, that is protein. So according to central dogma, what is the step? DNA will pass its information to RNA, RNA will take that information to ribosome, ribosome will make protein. Clear? So now this step which I am having, where DNA information goes to RNA, this step we call transcription. So what is happening in transcription? See, I am having a double stranded DNA like this. Now same structure I can show in ladder form in this way. Now whatever sequence there on DNA, suppose out of the two strands of DNA I am showing only one strand here. So on DNA sequence there, it is called sequence of nucleotide. If you see here, A, T, T, C, C, T, like this sequence there. So by using this nucleotide sequence on DNA, we have to make RNA. How to make it? For that, basic rules which there about pairing, we should know that A is always pair with U and G is always pair with C, or vice versa. If C there then G, G there then C, U there then A there then U. Remember that when T will come, T is present only in DNA. Just now we discussed. So when T comes, you have to make T is a optional there for U. So T there then A you have to catch as a complementary sequence. Clear? Now imagine that this is one DNA strand I am taking. Opposite strand which we make, that is our RNA there. Now see here, if on DNA red color is this latest DNA there, green color will make a new RNA. So here A there, then complementary to A, what there? U. Therefore U will come here. For T complementary, who is there? A. So T A will come. Again T A will come. Here see C there, then who is there? G. So C complementary G. C complementary G. T there, then A will be the complementary to that. So in this way, if I am having one strand of DNA, by using the nucleotide sequence of that DNA, I can make a RNA. This is my RNA ready there. So what we did here? By using the nucleotide sequence of DNA, we synthesized the RNA. So this synthesis of mRNA, any RNA you can catch. So synthesis of RNA as per the nucleotide sequence of DNA is called as transcription. So in this transcription process, main you have to understand that how to make the complementary sequence. Let's do it. Rules what there? Just now we discussed. G is always paired with C and A is always paired with U, where it is paired with T in DNA. Okay. Now suppose that I am having, this is a DNA one strand and I am going to make a RNA here. Can you tell the sequence for this? What will be sequence will come for RNA? Check. G. What is G complementary? C, sir. Okay, very good. If C there, then what will come, sir? G will come. Complementary to that. G there, then C will come. T there, then A will come. A there, then T. C there, then G. A, for A complementary, U there, so U will come. T there, then A will come. So here we made RNA. How? By using nucleotide sequence of DNA. This only process is called as transcription. So transcription is nothing but synthesis of mRNA, as per the nucleotide sequence of DNA. This process is called transcription. Now let's try here. You will help me. Now A there here. Imagine that this is DNA strand there and here I am having new synthesized RNA strand which we are going to make here. A there, then what will come here? Say with me. A there then. What is A complementary? U. Okay sir. U will come. C there then what will come? G. C. G. G there then C. T. A. A there then U. C. G. RNA ready. So synthesis of RNA as per the nucleotide sequence of DNA is called transcription. Clear? Now, if you see this whole process, to make one RNA molecule, how many strands are necessary? Only one strand, sir. If one DNA strand I am having, from that I can make RNA. This is only important point there. Even though DNA double stranded there, only one strand of DNA is used to make RNA. Clear? So now the whole process which we are discussing about transcription, 3 points are very important there in this. One, its definition. Definition that only, simple. What is the definition I am having here? What we are making here? mRNA. Any RNA you can talk. So mRNA is synthesized as per the nucleotide sequence of DNA. Here see. This is my DNA there. DNA is made up of 2 strands. This is blue color first strand there, green color second strand there. Both it is a 2 strands of DNA. Now out of that I am making, this is my mRNA there. This is my mRNA there. To make this mRNA I will use only green strand. I will not use only blue. Are you getting? Out of this 2 strand, to make mRNA I am using only green strand. So therefore I am telling that only one strand of DNA out of 2, only one strand is used to make RNA. Is it clear the second point? What is the first point? mRNA is synthesized. mRNA is synthesized as per the nucleotide sequence of DNA. Okay sir. Second, out of 2 DNA strand, how many used for RNA synthesis? Only one. So only one strand out of 2 is used. Now if you see carefully, that complementary rule just now we discussed, no? What is the complementary rule? A always pair with U, G always pair with C. This is complementary pairing rule there. Now if you see here A there, here also A there. That is same there, no? It is not complementary. Here see, U there, here U there, here T there, because thymine is present in DNA. G there, here also G there. So this strand which we called here coding strand, in that we are not interested only. Why we are not interested? Because this strand's information we not used. Information which is used to make the RNA is this green color there. So my mRNA is complementary to green. Complementary, understand? Complementary means opposite to that. So mRNA is complementary to which strand of DNA? The strand of DNA which is used to make that. Not with blue. With blue it is not complementary. It is similar there. If you read the sequence, only if you leave this T, same sequence you will find. Here and here same sequence there. So I am not interested in this blue strand. I am interested in green. Why green? Because from green only we made this. So the nucleotide sequence of mRNA is complementary to this strand of DNA. Clear? 3 points. 1. What is transcription? mRNA is produced as per the nucleotide sequence of DNA is called transcription. Now in your textbook, somewhat in that paragraph in different way it is given that. So this line is given somewhere up. This is called transcription is given at the last line like that there. So you carefully listen what I am telling. How to define transcription? Simple. mRNA is produced as per the nucleotide sequence of DNA. mRNA is produced as per nucleotide sequence of DNA is called transcription. 1. Second, only one strand of DNA out of 2 is used for RNA synthesis. mRNA sequence is complementary to one strand of DNA. This mRNA sequence is complementary to this green strand of DNA. Third and last, what difference you find? Instead of thymine, RNA is having uracil. This already we discussed that in mRNA or any RNA, thymine is not present only. So instead of thymine, what is present? Uracil. Is it clear? So here important concepts, what there? One, mRNA is produced as per nucleotide sequence of DNA is called transcription. Out of 2 strands of DNA, only 1 strand is used for making mRNA. mRNA sequence is complementary to 1 DNA strand. And last, in RNA, there is a uracil instead of thymine. Is it clear? Till here any confusion to you? Now this mRNA which is produced here, this mRNA where it forms, that is very important there. See, now already we understood that our DNA is present in the nucleus. If I am making the mRNA is made by using DNA only. Because DNA information only will come to RNA. Now where DNA present, there only RNA synthesis will happen. So in paper they can ask to you where transcription takes place. Transcription means DNA to RNA. This making process, where it takes place. So your answer should be nucleus. In nucleus only RNA will produce from DNA. Now once your mRNA is ready in the nucleus, it will come outside the nucleus into the cytoplasm. Why sir in cytoplasm? Recall the central dogma. This mRNA which comes outside the nucleus into the cytoplasm, it will go to one very important structure which we discussed, that is ribosome. Because our ultimate product protein, remember that ultimate product protein is made by ribosome. So this mRNA will come here to meet the ribosome. So making mRNA from DNA, it happens in nucleus. Once your mRNA is ready, this mRNA will bring the message in the coded form to ribosome in the cytoplasm. Is it clear? So mRNA's role, what there here? Whatever information that DNA was having, now it came on mRNA. mRNA will bring the message about protein synthesis to ribosome. And ribosome is a protein factory there. So as per the message of mRNA, ribosome will make the protein. Is it clear? So this is all about your transcription. I hope till here the concept is clear to you. If you like our videos, please subscribe the channel and share the link with your friends. Thank you.